All right, now let's see if we can finish up chapters 12 and 13. Um, so we talked about protein synthesis and this flow of information from DNA to RNA via transcription and then to proteins via translation. So again, the DNA is providing the ultimate instructions for the making of proteins, and of course the proteins are the molecules that carry out the work inside of cells for the most part. So um, in all of our cells, we have the complete genome. We have all 46 chromosomes, all 23 pairs throughout all the cells in our body. Um, but our cells, each cell does not make use of every chromosome well, they do make use of every chromosome, but they don't make use of every gene on every chromosome. That is, only certain genes are expressed in um, certain cells. So how does a cell know which genes need to be transcribed and translated, essentially which genes need to be used? And it comes down to these things we call transcription factors, which are these molecules, these compounds that, when present, basically are telling a cell that a particular gene needs to be transcribed. And so, as we saw, transcription is carried out by RNA polymerase. RNA polymerase, though, needs a little bit of help. Um, and what happens is when when the transcription factors are present, the transcription factors and the polymerase will form this complex that then bond to the promoter. And you remember we talked earlier about promoters or sections of DNA that are next to genes that essentially tell the polymerase this is the beginning of the gene, and transcription will proceed along the gene. No transcription factors, no transcription of the gene. So essentially, you'll recall we talked about how on a chromosome you can have, all chromosomes have multiple genes, and we don't transcribe every gene at one time. We only tend to transcribe them one at a time when the product of that gene, that protein, is needed. So you can imagine what we have are different transcription factors that are responsible for causing different genes to be transcribed. Now notice here, sometimes you can have a single transcription that might cause two genes to be transcribed, but those would be genes whose products both are needed to help carry out a process. All right, so transcription factors essentially allow for cells to become specialized. So for a nerve cell to become a nerve cell and only make use of the genes that nerve cells need to use, or your muscle cells or skin cells or cells of your liver or kidney or whatever. Um, so as you'll recall, of course, we all start out as a zygote or a fertilized egg, and then the zygote starts developing, starts dividing, and we develop into an embryo. Um, and at some point, the embryo, of course, consists of multiple cells. And so what happens is once you start to get multiple cells present, you start to get cell differentiation such that different parts of that embryo, different cells of the embryo have different transcription factors present. And so these cells begin to differentiate because they are expressing different genes. In this simplistic example, you can see this batch of cells are expressing transcription factor 1, and over here it's 2, and down here it's 3. And so this allows for the expression of different genes, the transcription of different genes, and cells to become specialized. Now, sometimes instead of um, Well, another way for cell specialization to occur is when we essentially um, block genes from being expressed or just turn them off. And so this phenomenon was known as RNA interference, is a way in which the expression of that gene can be blocked. All right, the way this works is that you produce these things called microRNAs. All right, we already saw messenger RNA, transfer RNA, and rRNA, but these are the different kind called microRNAs. And what happens is a piece of RNA that's produced is chopped into small pieces by this thing called the dicer enzyme. And those small pieces of RNA are then used to basically bind up messenger RNAs and to ultimately kind of break up that messenger RNA. So what the microRNA is doing is preventing a gene from being used or expressed by essentially disabling the mRNA that's made from that gene. So essentially, if the mRNA is no longer needed, this is a good way of disabling it and getting rid of it. Now, another sort of more permanent way to block gene action or gene expression is what's called DNA methylation. And what that is there are these things called methyl groups, which is a CH3. And when these methyl groups are added to the DNA, it essentially locks up that DNA and keeps it from being transcribed. So if you have particular genes that just are not needed in nerve cells and are never going to be used, then this is a way of just locking them up and keeping them quiet and them not being expressed. DNA methylation, it's a pretty common thing. All right, so now that's the control of the expression of genes. Now, mutations. So a change to the genetic information, what we would call a heritable change. And so by that we mean a change in the genetic information that can be passed on to from parent to offspring. So what kinds of changes can occur to our genetic information? Well, at most basically we have what we call point mutations. And a point mutation is when we take that sequence of nucleotides and bases in our DNA and you just make a simple single change to them, and they come in various forms. For example, up here, this is our normal or original strand of DNA, and down here we see some that have mutated. So for example, here, instead of having the sequence GCA, we now substitute a T, and you notice that changes the mRNA. The blue sequence here is the mRNA. Up here, GCA codes for CGU, and so that is the codon that codes for arginine. When we change that codon to CAU, notice it becomes histidine instead. So this point mutation, this substitution has the potential, it changes a single amino acid in our polypeptide. Now that may or may not have a significant change on the functioning of that polypeptide. But let's look at insertions and deletions. So here we're inserting an extra base between this C and G, and over here we're deleting a G. And so now we have an extra base or we're missing a base. And that causes what's known as a frame shift mutation. That is, you shift the reading frame in the DNA and then the mRNA and the codons. And so now instead of having AUG, CGU, ACC, UUU, we have AUG, but now we've got UCG, UAC, CUU, etc. So when the ribosome is moving along that mRNA, it's going to be reading different codons because of the insertion or the deletion. And notice this tends to change the amino acids, all of them, or at least a lot of them, going downstream. So instead of changing one that you get with a substitution, you now get a bunch more change. And so this is very likely to lead to a protein that's not going to function at all or very well. So frameshift mutations are pretty significant mutations. Now, on a larger level, you can get mutations of the, the whole chromosome, essentially, not just a single base here or there, but the whole thing. So, for example, you can have deletions where you can have sort of big chunks of the chromosome that are lost or deleted. You can have duplications so that now we have two copies of this particular segment or this particular gene, perhaps. Inversions where the portions sort of switch around. And then translocations where a part of one chromosome is translocated into a different chromosome. So part of your number 1 chromosome might translocate to a part of your number 2 chromosome. These tend to be pretty significant mutations and often bad, although not always the case. Sometimes duplications are not necessarily bad. You just are now, instead of having one copy of a gene, you have two copies of it on a chromosome. With inversions as well, they're all still present. They've just sort of switched around. Now, the effects of mutations. Now, sometimes they can be silent mutations. So, for example, if we look back here at our genetic code, notice, for example, that with glycine, there are 4 different codons that code for it. All it has to have is the first 2 to be G. The third one can be any of the bases. So essentially, if you have a mutation that changes the codon from GGU to GGC, you still get the same amino acid in your protein. So that is what we would call a silent mutation. It has basically no effect on the phenotype. Okay. But other times you can get mutations that change the phenotype, and those are the ones that can be significant. The protein can be mutated. It doesn't work very well. Mutagens, things that can cause mutations, radiation, exposure to X-rays, things like that. Exposure to ultraviolet light, in particular, can damage skin cells. The smoke from smoking cigarettes or any kind of tobacco products can be damaging to your lungs or your throat, the cells there, and cause mutations. Certain types of chemicals, which essentially smoke contains those chemicals, but other kinds of chemicals that you might be exposed to can also be, we would say, mutagenic and basically damage the DNA. Now, mutations. Are all mutations bad? Well, they can be, but not always. There are examples of mutations that have occurred that basically change the protein but don't make it defective, but perhaps even enhance the effect of that protein or just make it work a little differently. It still works. So mutations can sometimes be beneficial. Now here's examples of some significant mutations that have occurred in certain types of organisms, particularly plants, where polyploidy is where you have whole extra sets of chromosomes. So for example, we are diploid and we have 2 copies of each chromosome, but there are plants which have 3 copies, 4 copies, 6, there might be some 5s, 8. So for example, here is the typical kind of blueberry we cultivate, and it is a tetraploid, and this is due to mutations that occur. It has 4 copies of each chromosome. Bananas are triploid, or at least the kind we eat. Wheat is a hexaploid, has 6 copies. And strawberries, the kind we eat, not wild ones, but the kind we eat because they've been domesticated and modified, are octoploids. And so these are all essentially mutations, but they have led to beneficial foods for us. Okay, that's it.